binding of the growth factor or insulin are capable of initiating an intracellular phosphorylation cascade. And we said that's a very typical trick, regardless of the mechanism of signaling, hormonal signaling. It often begins with this phosphorylation cascade. It may involve protein kinase A, as we saw in the cyclic AMP pathway. But there are many avenues by which this phosphorylation cascade can be initiated. Now, you can see here they're just saying signaling molecules. That's your clue to understand there are many, many steps involved in the signaling pathway. We've learned that some intracellular signaling pathways, for example, culminate in exocytosis. We found, for example, that insulin is released by a signaling cascade that culminated in the calcium-dependent exocytosis of the hormone. But that's not what is happening here. These bizarre-looking things that look like lemon wedges in a little cell membrane or whatever. The implication here is that these are glucose transporters. These are intracellular glucose transporters. Consequently, when the cell receptors, we'll call them TK receptors, bind the insulin, that will initiate an intracellular phosphorylation cascade that culminates in mobilization. The other term that you'll often see is translocation of intracellular glucose transporters, not receptors, but transporters, to the membrane. So the glucose, the transport or mobilization of the glucose transporters is hormone dependent. It is insulin dependent in this case. And so once these transporters, these glucose transporters have been translocated to the membrane, at that point they're in a position to allow glucose to pass into the cell according to its concentration gradient. These are still passive transporters, but their translocation is dependent upon the insulin. And that's really the essence of this term, or this distinction up here when we talked about insulin dependent and insulin independent tissues. The GLUT4, which is the transporter we're looking at here, for the most part is an insulin dependent transporter. GLUT2, which you recall we encountered when we looked at the pancreatic beta cell, and we went through that sequence of events. That is a receptor that allows glucose to pass through the membrane, but it does not depend, its mobilization does not depend upon insulin binding. So be aware of that very important distinction. The major complication, though, the major complication to this simple dichotomy, in which we can send every cell in the universe into insulin dependent or insulin independent, is that you will find many cells have insulin receptors. Many cells will have insulin receptors, but they're not for the purpose of mobilizing transporters. So you maybe want to be thinking about why then would these cells have insulin receptors? And as you think about it, remember what insulin does. Remember the function of insulin and why the cell might find that information useful. And we'll come back to that in just a second. So just be sure, I can't warn you enough, I can't tell you how many times I've tripped folks up on trivial little test questions where they fail to differentiate these four terms. So make sure as you look at an illustration like this that you can fully and thoroughly and under all circumstances differentiate in particular the receptor from the transporter. And of course the ligand in this case would be the insulin, and what is the substrate in this model? Glucose. So as a rule, just again in the interest of time and to keep things rather simple, we're going to argue that for the most part, adipose or fat cells and skeletal muscle cells are the predominant insulin-dependent tissues. They utilize predominantly GLUT4 transporters, and again, these are the insulin-dependent transporters. And one would expect, therefore, that as the insulin binds to the membrane receptor, GLUT4 transporters are mobilized, that as a consequence, the intracellular glucose concentration would increase, and the cell that can then use that glucose, that fuel, that energy, to make things. And therefore, we argue or would argue that insulin is anabolic. It's functionally anabolic for these cells. And I mentioned a couple of the enzymes here. They're not terribly important, except we need to make sure that what cells often do with this abundance of glucose coming into the cell is they'll use it to put it away, put it in the bank for a while, store it away, either as glycogen, if it's an adipocyte, utilize that glucose to form triglycerides, for example. Now, I indicated a moment ago that we do need to deal with these troublesome cells that are not insulin dependent, but nevertheless have insulin receptors. And a classic example of this would be the hepatocytes. The hepatocytes, for the most part, utilize GLUT2 receptors as opposed to GLUT4 receptors. So they utilize GLUT2, but having said that, they nevertheless have insulin receptors. Of course, in the liver, you know that the liver is very important for regulating systemic blood sugar. And, for example, the liver has ample, copious glycogen stores, glycogen being a polymer of glucose that can be broken down as necessary to elevate systemic blood sugar levels. The liver also has, these hepatocytes in the liver also have the ability to undergo gluconeogenesis. And that's important because these cells they have the ability to generate glucose from non-carbohydrate precursors such as the lipids and the protein. So in the liver, again, we'll just sort of make this very simple, but in the liver then, you can see what insulin does. It tends to promote glycogen formation, right, from the glucose substrate. By the way, remember in your basic biology, you often ran across that glucose 6-phosphate. In fact, we encountered that in the beta cell in the pancreas. A lot of cells do this, but I want you to be, especially in the liver cell, think about why this particular sequence of events is so important. We know that glucose 6-phosphate is essential for glycolysis, the Krebs cycle, and so on. But that's part of what happens when hepatocytes bind insulin. When hepatocytes bind insulin, remember these cells are not insulin dependent, one of the things that happens is this sequence of events occurs. So that might give you some clue as to why the liver cell would have those insulin receptors. Insulin in the liver cells promotes decreased, it's kind of an odd way to say it, but reduces gluconeogenesis because, again, what is the insulin telling the liver cell? Hey, I've got plenty of glucose in the blood. You don't need to make any more. Put it back a notch. And then it also promotes storage of fats. Again, this is both direct and indirect. Other cells are doing this as well. But if times are good, if you have lots of food and lots of raw materials and energy, once again, all of the cells, including the liver cells, can be anabolic. So again, just to make no mistake, if I misspoke last time, I'm sorry, but insulin is extremely hypoglycemic, or it is a hypoglycemic hormone, and it's anabolic. Now, be sure you understand what we mean by hypoglycemic. It means that in that two-compartment universe that I postulated before, where you have cells in the blood, hypoglycemia implies that we're moving glucose from the blood into the cells. Before we leave this topic, I just want to make sure, because I've sort of beat around the bush here. I think I've given away the plot, but I just want to be sure that we all understand. Can someone tell me then why the insulin independent, or I guess that's really not a good way of saying it, the hepatocyte, which does not depend upon insulin for movement of glucose into the cell, nevertheless has insulin receptors? Somebody tell me why that would be. Yeah.
The secondary way to regulate blood sugar decreases gluconeogenesis. So when there's a prevalence of glucose already in the blood, it's not going to be pumping out more, and will.
It is or is not?
It's not.
Okay.
And with the glycogen formation, it will be pulling in more of the glucose that's already in the blood.
You're exactly right. I agree with you in terms of the final product. That's really all there is interested in. We're interested in understanding the big picture. So David is arguing that the presence of insulin in the blood is basically giving the hepatocyte directions, telling it what to do with its available resources and allocate its resources in a sensible fashion. The only thing I'm asking in addition to that, so when the insulin binds to the insulin receptor on the hepatocyte and ends up and achieves the result that David mentioned, what's the mechanism by which that occurs, I guess is what I'm asking. In other words, why do we need a receptor to do that? And if we go back to that previous illustration, I just want you to understand that insulin receptor is initiating a variety of intracellular signaling pathways, specific pathways, the biochemical pathways, which we don't have to get into. But those pathways culminate in the formation of glucose 6-phosphate. They regulate the rate of gluconeogenesis and so on, regulate the rate at which glycogenolysis occurs. What is glycogenolysis? Breakdown of glycogen. All right, so just be aware that that's so the hepatocyte insulin signaling does not culminate in mobilization of glucose transporters. Rather, it regulates the intracellular metabolism. And the insulin then is basically, as David points out, telling the cell how to behave under those circumstances. In most textbooks, you will see the relationship between insulin and glucagon as being sort of reciprocal or mutually antagonistic. And I think for the most part, that's relatively accurate. There's a problem with that. And I'm going to try to talk a little bit about that. But in general, you should know that glucagon released from the alpha cells of the pancreas. We talked about that before. These alpha cells produce glucagon in general when under two circumstances. And this is where things get a little complicated. And I don't want to get too deep into this. But glucagon is released from the alpha cells, number one, primarily in response to circulating amino acid levels. And that, we'll see why that's a little bit important. But number two, probably more important for our purposes in this course, in response to depleted or reduced glucose levels. All right. So, for example, when you're asleep at night or when you wake up in the morning, your glucagon levels are going to be elevated, thereby telling your and that glucagon is going to bind to hepatocyte receptors and tell the hepatocytes to increase gluconeogenesis, increase glycogenolysis, and thereby increase the circulating blood sugar level. So again, keep in mind the primary effects of glucagon are on the liver, and you can see a number of the processes that are stimulated by binding of glucagon. Glycogenolysis, breakdown of glycogen, depolymerization of glycogen, increasing the availability of glucose in the blood. Gluconeogenesis, again, an alternate mechanism by which blood sugar can be elevated by these specialized cells. And then ketogenesis. If it's true that blood glucose levels are low or depleted, that could be bad news for cells because they need that energy to perform all the moment-to-moment, day-to-day work they have to perform. So if you don't have adequate stores of glucose and they're not available in the blood for a long period of time, what can the body do to sort of compensate? Right, basically resort to the utilization of the fat stores. And when that happens, if you know anybody, I think some of you said you might be on the Atkins type diet or ketogenic diets. Ketones are produced, and these are products of fat metabolism.
When someone gets A1C measured, what exactly is it?
That's actually a sort of that's a little complicated, but it's basically just the residual consequence. What happens to a red blood cell when there's a fairly elevated amount of glucose in the blood over a long period of time? So it's sort of that you'll have various measures in your toolbox. for assessing whether a patient is hyper or hypoglycemic, potentially diabetic. But that's just one marker. And generally it's a marker, say 3-4 weeks of glucose levels in the blood. So by ketogenesis, we're just implying that the cells are moving away from glucose utilization to lipid utilization, and that type of metabolism has a number of consequences. First of all, not all cells can use ketones. The brain can use them, the heart can use them somewhat, but not all cells can. And then there are some other side effects. You've probably heard of ketone breath. What is that? Well, it's more the smell of nail polish because it's the acetone and the butyric acid that's present in the breath as a consequence of metabolism. But those might be indicators of perhaps, for example, a person with diabetes who cannot utilize glucose. So glucagon indirectly, therefore, acts to elevate the blood sugar mechanisms we've already pointed out. As I say, there's one, I thought there was a complication. I thought I had it up there on the slide. Oxidation of lipids, formation of ketones. Again, we've already talked about that. I was going to talk about the amino acids, but if I don't have to get into that, we won't worry about it. In general, maybe you can give me the standard textbook explanation. Why would glucagon be released when amino acids are elevated? Why would amino acids, I guess is a better way of saying it, why would elevated amino acids trigger release of glucagon? What is one of the important functions of glucagon? The hepatocyte might utilize the amino acids for? Gluconeogenesis, yeah. And so in high protein, if a person has let's say a consistently elevated or is on a high protein diet, you might find both insulin and glucagon levels elevated temporarily. Just a quick, before we leave, you should know a little bit about diabetes. This is not a pathophys course. You should understand that it used to be quite fatal. It used to be quite a serious condition. You should know the difference between type 1 and type 2 diabetes. In type 1, there's autoimmune destruction of the beta cell, this progressive destruction, and a lot of factors affect the rate at which this occurs. But the net result of type 1 diabetes is just an insufficiency of insulin. So if you don't have insulin, what's the problem? Yeah, you're going to have two major problems. You're going to go into wasting in the absence of any therapy because the cells they cannot utilize the fat cells, the muscle cells, and so on, cannot utilize the sugar that's present in the blood. And also, we say that these patients exhibit a number of signs, including glucosuria. Glucosuria, which is what? Yeah, glucose in the urine, which implies there's so much glucose in the blood, even the kidneys can't handle it. And my point there is that glucose can act as a toxin. It can actually have significant toxic effects in many tissues of the body. So there are a number of consequences of insufficient insulin. In type 2, the patient may be even hyperinsulinemic. They may not be, but they may be hyperinsulinemic. So they have plenty of insulin. They may have plenty of glucose, but what's the problem in type 2 diabetes? Signaling or receptor issue. We normally say that there is a receptor problem, and oftentimes the receptor is working fine. It's just the intracellular signaling mechanism. Once the insulin has bound the receptor is deficient. So in type 2, there are all kinds of therapies that can be addressed. When we talk about the GI tract physiology, there was a very interesting study that was reported last week regarding how this may be addressed in the future. I was very surprised at the finding, and we'll defer that discussion a little later. It says your cause is probably intracellular. I think you're going to be very frustrated if you actually try to find the intracellular cause of all types of diabetes, type 2 diabetes, because it's probably variable, and a lot of different therapies can have differential results for different patients. Actually, I'm going to go to this one for you folks.
Insulin resistance is a condition in which the body cannot use insulin efficiently. About 60 million Americans have insulin resistance. Insulin is a hormone produced by the pancreas that instructs cells to take up glucose from the bloodstream when there's too much glucose in the blood. In insulin resistance, cells of the body do not respond to insulin efficiently to remove glucose. The pancreas responds by making more insulin to try to keep the blood glucose levels normal. The insulin-producing cells of the pancreas slowly become defective and eventually reduce in number. As a result, blood glucose levels rise over time, causing diabetes to develop. Insulin resistance is a core metabolic disorder associated with type 2 diabetes. 1 in 4 people develop type 2 diabetes, which can increase the risk for heart disease and stroke. Risk factors for insulin resistance. Family history of type 2 diabetes. Race or ethnic background of African, Latino, or Native American descent. Being overweight. Hypertension or high blood pressure. Increasing age raises risk. Previous gestational diabetes or delivering a baby over 9 pounds. There are often no signs or symptoms of insulin resistance. If you have a mild or moderate form of insulin resistance, blood tests may show normal or high blood glucose and high levels of insulin at the same time. One test does not usually confirm insulin resistance, but a combination of tests can. Fasting insulin blood levels can be measured by taking a blood test after fasting for 8 hours or overnight. Glucose tolerance can be measured using either a fasting plasma glucose test or an oral glucose tolerance test. A physical exam and medical history combined with insulin and glucose blood tests can help your doctor determine if you have insulin resistance. There are a few things you can do that may help prevent insulin resistance. Engage in physical activity for 30 minutes a day. Physical activity can help your body's cells process insulin better. Lose 5 to 7% of your body weight if you are overweight. Eat a heart-healthy diet that can help you lose weight and lower your risk of diabetes.
That point about I thought all babies were over 9 pounds now. So, all right, that's all I think. The other two, I think, are a little bit elementary for you. All right, any questions then about the pancreatic regulation? We'll move on then to a discussion of reproductive physiology. In most healthcare or medical curricula, reproductive physiology is normally considered part of endocrine physiology. So you want to make sure you appreciate the very complex interplay of hormones that we'll be talking about, some of which we can clarify, some I'm not sure we can. But this first unit that I want to spend a little time talking about, we can call it, I don't know, reproductive genetics. And I have to admit, I'm often stunned when I talk with some healthcare professionals, even physicians, about the misconceptions and assumptions made about genetics of reproduction. Now, this first slide, I'll leave it to you to look through, but I want to make sure you understand that there's a distinction, a very important distinction between, for example, sex and gender. We're not going to get into gender here. We're not going to get into psychology. Professor Coombs may take that up with you in his discussions of the human mind. But I think you're going to find that just the biology of sexual reproduction is sufficiently complicated to keep us pretty much confused most of the time. But do understand that this distinction is very important. The biology is certainly going to have a bearing on the psychology and vice versa, but that's not our province in here. We're just going to talk about the biology and sort of work through some of the concepts and notions that are relevant to the biology of reproductive genetics. So just general principles. You know that for the most part, every cell in your body has 23 pairs of chromosomes. That's not true for all cells. You have red blood cells. You get some cells that are multinucleate. We talk about those crazy osteoclasts, for example. The liver seems to be fond of genetically bizarre cells. But in general, we say that your body, your tissues are made up of diploid cells. Diploid meaning there are 23 pairs of chromosomes, half of which you inherited from one parent, half from the other parent, at least biologically. The first 22 pairs are called autosomes. And they're called autosomes, the actual etymology isn't too important, but they're called autosomes because they have the ability to exchange genetic material during their creation, during what we're going to call meiosis. The 23rd pair of chromosomes are the sex chromosomes, and their ability to cross over or recombine is very limited. We'll talk more about that in just a moment. As I say, the sex chromosomes, the 23rd pair, properly speaking, these consist of at least in humans. Other mammals have some more bizarre setups, but in humans, it's either X or Y. The normal females would have two X's. A normal male would have X or Y. And I have to put the word normal in parentheses because I'm not sure we're in the business of arguing who or what is normal. The X chromosome is quite large. You can see it contains about 1,000 genes. Can't get along without it. I think the largest gene in the human genome is on the X chromosome, if I'm not mistaken. The dystrophin gene. Y, that's ridiculous. It's perfectly superfluous. You don't need it at all. So you can see I have 80 genes there listed. That's really giving it the benefit of the doubt. And what would the function of those genes be, do you think? The Y chromosome. That's ridiculously silly. What's a Y chromosome? Yeah, chiefly for male characteristics. And if male characteristics are important, why wouldn't all humans have them? Right? So it's just a few silly male characteristics. Now, curiously, on the X chromosome, there are also genes for normal male development. But we'll talk about expression in just a moment. So even at this early stage, and then I'll be back to torment you in some other classes after we finish this first semester, but we'll come back and talk about genetics a little bit in some of the other classes. But you should be familiar with the notation used to describe, and we'll call this karyotype. We'll talk about a karyotype in a moment. So again, the normal female would include 46 chromosomes, 23 pairs, and we indicate the sex chromosomes after the comma there. The male would be 46 chromosomes XY. Now, you began as a single cell, a single diploid cell. The single diploid cell, we could call it a zygote, and that zygote was formed through fusion of two haploid gametes. So, for example, we're going to call the female gamete the oocyte. We have to be careful with our terminology. And so note the karyotypic designation here, 23X. Spermatocytes would be the male gamete, and you'll notice the spermatocyte can carry an X or a Y, so it's really the sperm that determines the sex, or at least the genetic sex. We have to be very careful with our wording here, because I think you'll find that I think I convince you that the terminology is inherently problematic. But we'll say that the spermatocyte is designated either 23X or 23Y. And as we've indicated, when the two gametes fuse, they form a diploid zygote that will then undergo a succession of mitotic divisions, forming morula, blastocyst, and so on, that progressively differentiates into three fundamental germ layers, and eventually a fully differentiated human being. But understand that when we're talking about normal diploid cells, that represents a genetic contribution from both parents. Of course, you now have to deal with the three-parent problem. It's approved in the UK. It probably will be approved here very shortly. Anybody know how you can have three parents genetically? You will. But it complicates things. Yeah. All right, so let's work through some of these terms. I'm not sure we're ready to address the three-parent problem now. Let's work through some of these terms to make sure we all have a common footing. So you're in the literature, my goodness, I don't know if it's our culture or a society or civilization or whatever, we come up with these crazy terms. They change every few years, and I don't know what to tell you except let's just go with the current lexicography as best I understand it. So by in the medical literature, when you see references to determined sex or sex determination, we're talking exclusively about the genetics, the genes. So that would be XXXY, XXY, XXXY, whatever. All right. So determined sex means nothing more than the chromosomal complement or some genetic variant thereof. And so that would, the determined sex would not be equivalent to gonadal sex or phenotypic sex. What would you think we mean by gonadal sex? What are the gonads? Yeah, testes and ovaries. So by gonadal sex as a first approximation, let's just argue that we're talking about the plumbing. Does the individual have male plumbing or female plumbing? And then we have to get into the whole issue of whether it's functional or not. But if, and then we need to talk about how either one or the other arises. But when we talk about gonadal sex, we're talking about does the individual have gonads or testes? And again, that doesn't have anything to say necessarily about whether they're functional. What is phenotypic sex? Yeah, what they appear to be. And you can see that might get into gender, which we're not going to touch in here. I just want to make sure phenotypic sex would be the outward appearance, and I think that might be a source of surprise for you too. But make sure you understand these are three different concepts, and we haven't even exhausted the possibilities yet. Now, with respect to the basic genetics that we should be familiar with, there's a mention here of homologous chromosomes. I've indicated that you have 23 pairs of chromosomes. For every chromosome, you should have a mommy chromosome and a daddy chromosome. Probably shouldn't be pointing to the colors because that would be sexist. And we might imagine in the room here, this would be the maternal chromosome, and this would be the homologous. What does homologous mean? Yes, yeah, similar to. So this would be the mommy chromosome, and this would be the daddy chromosome. And along that chromosome, we can just go down, you would have an allele. So this would be the allele, which is make something up, hair color. All right. So this would be, what did I say? Maternal allele for hair color. This would be the paternal allele for hair color. They code for the same thing, generally speaking, but they may not be the same gene. This could be blonde. This could be for black hair. And hair color is not that simple. You see what I'm saying here? So we call this the allele. So there'd be an allele here for, I don't know, hair color, finger length, whether you're a knife, good or bad, and so on. So we would have a different allele arranged in more or less the same sequence along the chromosome. So again, just be aware that, of course, the gene, when we talk about the gene, we're talking about the specific sequence of base pairs that codes for a given, for now, rather than say protein, let's just say RNA or characteristic. So are we familiar with the idea of homologous chromosomes? This is going to come up again a little later because we understand meiosis, which again, we're working through today. You need to understand this concept of homologous chromosomes and an idea of alleles as they are present in the homologs. Again, each homolog from each parent. What do we mean by gene expression? What is gene expression? So if this is the gene, if this is the maternal gene for hair color, how would that be expressed? Yeah, I mean, we could be, that would be the characteristic. We could say it could be for a protein pigment or some sort like that, or the type of pigment or whatever. But you should understand that properly nowadays, expression is just called, the better term is gene product. And the gene product is usually a protein or just RNA, protein or RNA. It doesn't have to be a protein because we found out that RNA does a lot of things we didn't previously believe. So a given sequence of base pairs in a stretch of DNA that we define as a gene is going to code for an RNA transcript, which may or may not code for a protein. And that is gene expression. All right. We're going to look at a little video in just a moment in which the narrator explains the actual shape, intracellular shape of these chromosomes. They don't look like the little X-shaped things that we normally think of. But let's say I'm a polymerase, and I'm going to be copying. Instead of having the genes run this way now, I'm going to have them run this way. So I'm a polymerase. I'm a fairly lazy polymerase, as you know. And I'm coming down here, and I'm copying the DNA into RNA. All right? Who's going to get copied first, and who's going to get copied most readily? The folks nearest me or the folks farthest away from me? Yeah. So what I'm saying is, the concept of epigenetics is this heretical notion. I say heretical because if any of you ever studied Lamarck in high school biology, doctrine of acquired characteristics? Why does a giraffe have a long neck? And so the giraffe, he started eating leaves in a tall tree one day. He ate all those leaves. Has to reach for higher leaves, and the neck gets a little longer, right? And then so his neck gets longer by the time he gets to be an old man like me. And then the remarkable thing is he's going to pass that long neck on to the offspring, right? That's the theory of acquired characteristics. Anybody see a problem with that? Does it work that way? Does nature work? No. We all say no because who overthrew that idea? Yeah, Darwin and Mendel and the notion of the traditional Mendelian genetics. But Lamarck was right. There are acquired characteristics, and you do pass them on to your offspring, and they pass it on to their offspring. So, but before we get into that, I want to make sure you understand the mechanism by which that occurs. And this is called epigenetic modification. And all we're saying is that in epigenetic modification, the genes, the base pair gene sequence, the APGC, does not change. That does not change. So when we talk about the new Lamarckianism, we're not saying that the sequence of the gene, the base pairs in the DNA change. What I'm saying is two things can happen to alter gene expression. What's the definition of gene expression? Make an RNA or a protein. Two things can happen. Number one, the chromosome changes its packaging or is packaged in such a way that certain genes are more accessible to the enzymes than other genes. Again, I'm walking down here, I'm gonna copy these first, or at least I'm gonna copy these most readily. I may never even get to Louise. Okay? He may never get expressed at all. And the other way this happens is through chemical changes. And I gotta be careful what I mean by chemical. Usually the most common example is by methylation, adding a CH3 group to those base pairs. All right? So what we're saying is that gene expression can be altered. You can alter your gene expression today. How do you do it? You get exposed to stress. You eat lots of food. You eat food with lots of methyl groups in it. Believe it or not, that's been shown at least in animals. Are you prone to stress? Do you think you have any stressful life? This was first discovered in the Scandinavian population who wanted to win a famine back in the early part of the 20th century. And what they found is, and we'll get off this topic very shortly, what they found is that the expression of genes was altered in that group of people that experienced that famine. And here's the important thing. That change in their gene expression was passed on to the next generation and the next generation. Exposure to insecticides, exposure to fast food is about the same thing, right? All of these things can alter gene expression, and this is the Lamarckian component, and that's why once you know the term, can be passed on to successive generations. So the last thing I will say on this topic is that there is a type of epigenetic modification that is sex chromosome dependent. There is a type of epigenetic modification that is sex chromosome dependent. By that I mean that if the gene arises with the egg, the genes either are or are not expressed Whereas if they are, let's just say they are expressed, if they arise on the sperm, they're not expressed. Okay? So that's called sex-dependent epigenetic modification. It's something you should be aware of because it's just in the news every day these days. We're looking at something here on the right called karyotype. All the karyotype is, at least at the level of our discussion, is just, we're going to take all the chromosomes, all 23 pairs of chromosomes, arrange them by size. You see the largest, number one is the largest sex chromosomes at the end here, and then I'm going to stain them. Now, staining, there are all kinds of stains, and the stains can be utilized to show different features. But I guess my question for you is, why would this be useful to you as a PA? What type of information could you gather from a simple karyotype like this? What do you mean by trisom?
21, 3, 18,
Yeah, so what you could tell from a karyotype is whether or not the individual has too many or too few chromosomes, right? And William is right, to 21, that would be trisomy 21 or Down, the type of Down syndrome, 13, 18, sometimes it would rise to trisomy. What is this individual? What's the sex of this individual? All right. Sometimes this happens. What would the sex of this individual be? Get rid of the Y. That's monosomy X. Monosomy X, that's Turner syndrome. That would be phenotypically female, but they're not reproductive female. So yeah, and what sometimes happens is one of these chromosomes will get tired of living here and hop over onto the 14. Sometimes the chromosomes will form circles. Sometimes the chromosomes will, a portion will invert and attach somewhere else. So if there are major chromosomal abnormalities, translocations, abnormalities in number, that can show up in the karyotype. So just be aware of what we mean by karyotype. And as I say, there are different types of staining that can help to bring some of this information out.
What we're seeing here is an illustration of a single cell, and we're slowly traveling towards its nucleus, the hub of genetic material. Inside, you can see chromosomes, the familiar X-shaped blob of DNA. Chromosomes don't really exist like this in most cells. In fact, this is only a fleeting structure. Most of the time, chromosomes look like this. These are 4 typical cell nuclei in which we've stained all of the DNA with a blue dye and one of the chromosomes in green. You can see it looks nothing like the familiar X-shaped cartoon structure of chromosome. Using a new technique that we've developed, we've created a much more accurate picture of how the DNA folds within the chromosome. The DNA is represented here by the single strand that weaves through the 3D structure. This is how DNA exists within a chromosome in its usual state, the state in which all the important functions of the genome are operating and controlled. 3D models of chromosomes allow us to map specific genes and other important genomic features onto the structures. Putting DNA into its proper context like this is important because the folding of DNA and the positions of genes on the chromosome contribute to genome control.
So as you look at this, I want you to remember what he said there. Now, it's true that during DNA replication, the chromosome doesn't look like this. It assumes that more linear shape. But this is the way your chromosome is right now. And of course, you would agree, most of you would agree, that your chromosome, your DNA is being used to direct production of proteins, all kinds of things. All right. And this is crystal or something here, and Louise is way down in there, one of those black areas, which is more likely to be expressed, the gene out here. So what we're saying is epigenetics is a function, at least in part, of the location of the gene in this very, very complex amalgam. All right, we'll stop there and take a break.